Hi everyone, my name is Taylor and I'm a PhD student in Hal Whitehead's lab. And today I'm going to be talking about a new method that my co-authors and I recently developed, which uses what we term identity calls to detect structure in acoustic datasets. Acoustic analyses can be powerful tools for illuminating structure within and between populations, especially for taxa that are cryptic, such as many species of frogs, or difficult to access, like marine mammals. Animal calls and repertoires are often compared using various measures of similarity calculated over a range of call parameters. For some taxa, however, the presence of group-specific identity calls may more clearly delineate various levels of biological structure than aggregate similarity measures. This is especially likely to be the case if those identity calls have been produced by selection pressure for signaling identity. Signal detection theory predicts that calls with such functions will exhibit features that enhance their transmission, like redundancy, stereotypy, and distinctiveness. We know that such group-specific calls exist for a diversity of taxa, a few examples being vampire bats, red wolves, and sperm whales. I specifically study sperm whales, and in social situations, sperm whales communicate using Morse code-like patterns of clicks called codas. Different cultural subpopulations of sperm whales, which we call clans, use different coda dialects. So the overarching goal of my PhD thesis has been to determine how sperm whale cultural dialects vary over space and time. But a persistent bottleneck on my research has been this question of how best to determine which clan made each coda if you're just given an acoustic data set. With that in mind, we began designing a new method for detecting structure in acoustic datasets, like my sperm whale dataset. And this method focuses on group identity vocalizations instead of on aggregate similarity measures of calls. In this new method, we first divide calls into repertoires. This can be done by individual or field site or year or day, depending on the dataset. Then we classify calls into types using contaminated mixture models. This type of mixture modeling looks for clusters in datasets using multivariate contaminated normal distributions, which is just a long-winded way of saying using a two-component normal mixture, where one component represents the good observations in your dataset and the other represents the bad or the noise. And this is a nice modeling approach because it lets those bad points be downweighted when calculating the component means and covariance matrices. After calls are classified into types, we hierarchically cluster repertoires of calls based on similarities in call type usage. And so this results in a dendrogram where closer repertoires use more of the same call types. Lastly, we look for identity clades and identity calls in that dendrogram. So identity clades are essentially clusters of branches in the tree that use certain call types, which we call identity calls, a lot more than any other part of the tree. And the thought here is that those identity clades may tell us something about the underlying biological structure in the animals we've recorded. At each stage, there are a number of parameters and settings that must be established. For example, during call classification, the user can set the initialization strategy for the classification, as well as the information criterion for model selection. And in the identity clade and identity call step, there are two parameters unique to the method. One being the critical factor, or crit fact, which is just how much more a call type is used in one part of the tree than another for it to become an identity call, and the minimum number of repertoires required for an identity clade to form. So we conducted trials to determine how reliant the final dendrogram results were to different parameter and setting combinations. As I mentioned earlier, this work was motivated by challenges in sperm whale research, but the underlying theory we think is potentially applicable across a diversity of taxa. So we also tested wren and cricket acoustic datasets to see if the program could successfully pick up on different levels of biological structure across the three. Jumping right into the results, here is a dendrogram of repertoires that were recorded from Atlantic and Mediterranean sperm whales. And so each branch is a repertoire, and we can add this heat map where each row is a different coda type. And the heat map shows how much that type is used by each of the repertoires. The last thing the program does is it overlays some color onto this, where the 3 different colors here are 3 different identity clades. And the plot on the right side is showing the coda types that are being used by those different identity clades. So we have in the color the identity clades, and on the right, also colored, the identity calls. And what we found for this data set was that across parameter trials, we got the same 3 divisions, which actually correspond to 3 known sperm whale subpopulations in this data set, the Mediterranean clan and 2 Eastern Caribbean clans. We did the same analysis for Pacific sperm whales, and we actually found evidence of 5 identity clades in this tree, 4 of which correspond to previously known cultural subgroups called the short, 4+, +1, and regular clans, but also a 5th clan, which hadn't been documented before. But now that we've expanded our data set even beyond what you see here, we're really confident that this is a new clan, which is exciting because it suggests this method is picking up on clans that previous methods did not. But I mentioned that we did these parameter tests, and this was actually the tree that varied the most from run to run when parameter values were changed. But one thing we noticed is that almost all of that variability seemed to focus on this one clan, the short clan. So a lot of trees would produce one clan, as you see here, but sometimes it would be divided into 2 or even 3. And while this might suggest that the method isn't working great, we actually think it's hinting at something more interesting, which is that there's more underlying variability or uncertainty in the codas produced by whales in this clan or clans. For the wrens, the program robustly delineated the 2 known subspecies, which was exciting. And similarly, for our field crickets, the program robustly delineated the 2 known species. So, going straight into the method's strengths and limitations, we were really excited that the method worked on these 3 very different case studies, because it showed that it could not only detect sperm whale subpopulations, but also wren subspecies and cricket species. And these 3 taxa use very different types of vocalizations. Another strength of the method is that it's customizable, so users can set parameters and settings based on features of their data set or aspects of their research question. For example, users who want to be really conservative in the identity clades and identity calls they find can increase that critical factor parameter or the minimum number of repertoires parameter to achieve higher conservativeness. There are a few limitations of the method, one being that we can't cluster calls of different dimensions at the same time. So this means that we wouldn't be able to compare sperm whale 3-click codas to 4-click codas directly. And additionally, there was some variation in the final results for certain data sets with different parameter settings and values. So this means that the program may not always provide a definitive answer about how many acoustic subgroups are in a data set. But again, we think that any variability users are seeing in their results may actually reflect real biological variability in the data that they have. It's worth stressing here that the presence of identity calls, like those that we found in these 3 data sets, doesn't necessarily mean that those calls are meaningful to the animals themselves. Playback experiments are a key next step to test if specific calls are used by animals to broadcast or infer identity. And we think this method can inform such studies by identifying calls that are potentially characteristic and likely to be perceptually salient to different population subgroups. We think this method can also aid conservation and management by starting to quantify acoustic diversity and detecting putative structure in acoustic data sets. And this is exciting because acoustic data, especially for researchers like us who study marine species, is often the cheapest and most minimally invasive data that we can collect. So I want to say thank you so much for tuning in and watching this talk, and I also want to thank the people and organizations on the screen who helped with everything from listening to sperm whale files to getting hands on wren and cricket data sets or helping with coding the program. And I'm happy to take any questions you may have.